It's a pleasure to be here this morning and thank the organisers for the opportunity. I'm going to take the short session I have this morning to describe work that's gone on over the last 6-7 years, and it's all been in collaboration with colleagues at the University of Kent and has culminated in the last couple of years in the development of the viral pseudotype unit. Now, our aim of this unit is to use a technology that will allow us to develop virus-like particles that are in essence harmless, but will allow detailed surveillance and serological studies of highly pathogenic, neglected and emerging viral diseases. When we first came to this project, or we first came up with the idea, the questions we wanted to answer were, was it possible to pseudotype some of these simple RNA viruses? Pseudotypes, or viral vectors, have been used widely in gene therapy applications on a limited number of viruses, but was it possible to adapt this technology and to enable the study of, as I said, emerging and neglected viruses? Could we then further manipulate or engineer this platform to allow this assay to be adopted by a much wider spectrum of laboratories? And how does it compare? You know, we come up with these viruses, we come up with these assays. Are they as sensitive, are they as specific as existing wild type or live virus neutralization assays? This is what we were aiming to answer, and hopefully we'll have some of these for you in the next 10 minutes or so. Given the background of the audience, I'm going to largely skip over introducing each virus as we come onto it. But I thought, so we're all starting from the same page, I'll give an introduction to pseudotypes. As I mentioned, these are effectively harmless viruses. They are non-replication competent. They can undergo one round of infection. They are engineered to contain a lentiviral core, and within this core there is a reporter gene that allows you to track the infection. That is the only genetic information within these virus particles, hence they can't replicate, and so once they're infected, you can just monitor for the reporter gene. The reporter gene is one of the mechanisms through which we've adapted these virus particles. The other is through incorporating different envelope glycoproteins. For instance, rabies, lyssaviruses, chikungunya, dengue, these can all theoretically be incorporated into pseudotype particles. So we end up with 3 plasmids. One containing the genes we need to form our viral cores. The other has our envelope glycoprotein, so this will end up studied on the viral membrane of the virus. And third is our reporter plasmid or transfer plasmid. Now, this has been genetically engineered to contain the packaging signal, so as I mentioned, this is the only genetic information within these virus particles. We take these 3 plasmids, transfect them into producer cells, and harvest the supernatant, which contains our viruses. So if we look at these cells in more detail, here, if we stain the cells with, this was with a Antirabies monoclonal antibody, you can see this is very specific membrane staining. Intracellular, we've got the GAG and the reporter staining. And when you take the supernatant, you dry it onto slides and you stain it for rabies with the rabies monoclonal, you see specific virus particles containing or incorporating these rabies glycoproteins. So the viruses harvested from our supernatant can then be taken on to use to study specific individual genes through stable integration, reporter or receptor studies, looking at specific interactions between the viral glycoprotein in the cell, and for what we hope will be surrogates to using live virus, highly pathogenic virus in neutralization assays. I started by looking at rabies viruses. Rabies is a member of the Rhabdoviridae, a number of plant and animal viruses, causes in excess of 55,000 human deaths each year. The vast majority of these are from infected dogs biting individuals, and quite scary statistics I always find when I read it is that over half of the world's population live at risk of becoming infected, live in rabies endemic area. If you're unlucky enough to become infected, become exposed, you do stimulate a humoral and cell immune response. However, this isn't fast enough, it's not robust enough, so we need intervention strategies to eliminate virus replication. If you do not receive these, symptoms will develop within 2 to 12 weeks. There have been longer incubation periods. Generally start off with flu-like symptoms which increase in severity all the way through to hyperphobia and cerebral dysfunction. But as I mentioned, we are lucky enough we have very efficacious vaccines against rabies. These will protect against the vast majority of circulating rabies viruses. And also, if you're exposed without receiving a pre-exposure vaccine, you have post-exposure prophylaxis in the form of rabies immunoglobulins. We picked the most commonly used rabies virus in diagnostic terms, CVS11 challenge standard virus 11. We cloned the envelope reporter gene and transfected it along with the other required genes to create CVS11 pseudotypes. And running neutralization assays with over 500 samples and plotting the neutralizing antibody titer on the y-axis here against the neutralizing antibody titer with the live virus assay, the FAVN, you can see we get very nice correlation and high sensitivity and specificity. We've gone on and developed pseudotypes to include at least one isolate from each of the 11 lyssavirus species. Here's rabies virus, it's one of the lyssavirus species. These are delineated into phylogroup 1 and 2 based on pathogenicity and neutralizing profiles. Antibody against phylogroup 1 viruses won't neutralize phylogroup 2 viruses, and vice versa. We've utilized this to try and identify epitopes within the lyssavirus glycoproteins that are important, or the most potent antigenic epitopes within these glycoproteins, to see if we can incorporate this into one glycoprotein and create this highly potent, broadly neutralizing immunogen. But that's a little bit on more advanced uses of pseudotypes. What's more pertinent, I think, to today's audience is the flexibility that we've introduced to this assay by incorporating a number of different reporter genes, LAC-Z, GFP, luciferase. Depending on the lab's requirements, there's cost advantages and time implications for each of these reporters. You can select your ideal candidate. We're currently working on introducing further reporter genes that will hopefully be cheap and fast. But that's definitely a work in progress at the moment. So it's not only lyssaviruses that we've been looking at. Influenza, Nigel Temperton, University of Kent, has been sort of spearheading this approach. We've cloned a vast number of HA glycoproteins for different influenza subtypes, both lineage A and B. I suppose this should now read H18 as of a couple of days ago, but as you can see, we've got a large library of influenza pseudotypes that we can use and have proved to be equally sensitive and specific. Further to this, we've incorporated the spike proteins of the SARS coronavirus, again work done by Nigel. When you compare neutralizing titers using the pseudovirus assay against the live virus assay, there's this nice correlation. More recently, we've been looking at the MERS coronavirus that was mentioned in the previous talk has been a public health concern over the last year or so. Other viruses that we're currently working on, which may be of interest to the audience, in collaboration with the Cambridge Infectious Disease Consortium, we're looking at paramyxoviruses, namely Nipah. Also the flaviviruses, Simon Scott and Stuart Mather at the University of Kent, looking at Japanese encephalitis. Collaborators, Barney King and Janet Daly at the University of Nottingham, looking at West Nile virus. Also to this, there are reports of dengue virus being successfully pseudotyped. Very low titers, very difficult to reproduce, possibly down to the extracellular budding versus intracellular budding nature of Flavies versus some of the more successfully pseudotyped viruses. And more recently, we've moved on and we're interested in looking at bunyaviruses, so Schmallenberg virus. Can that be pseudotyped? Emma in the lab is hopefully working on that at the moment. There are other viruses, sort of Rift Valley fever, that we also have our eyes on. But these are all viruses that are important public health concern, and the serological study and surveillance of these viruses can be vastly improved by using pseudotypes. There is the standard pseudotype neutralization assay. You make your virus, you run it with the sera you want to test for antibodies, and you get a readout. You can do that with another virus using a different reporter. But to save money, save time, why can't you just use two different reporters in the same plate? So both viruses added to the same plate will enable you to undertake the same experiment, but at less cost and much faster. Proved a good approach in the preliminary studies we've done, looking at single neutralization titers versus when you add both viruses at the same time, both for lyssaviruses, Lagos bat virus and Macola virus, and also when we look at two flu viruses, H5N1 and H7N1. Not only is it the reporters that make this assay nice and flexible, it's also the stability. Obviously, the areas where the major burden of these viruses are resource-limited countries. The cold chain may not be able to be maintained. When we look at the rabies virus CVS11 and the influenza virus H5N1, you can see when you store these viruses at room temperature or at 4 degrees in the fridge, they're stable for a matter of days, weeks, which was quite surprising to us. So if you do have a breakdown in the cold chain storage, they'll be able to survive. But obviously, if you thaw the virus and refreeze it, what happens to the virus titer? Here we have flu H5N1, CVS11, the rabies virus, and this is HIV pseudotype. Both the flu and rabies pseudotypes lose around 3% of virus titer each time they're freeze-thawed, so you're not losing much of the virus titer, compared to about 9% each time for HIV. And lastly, we've got the latest development by Stuart, University of Kent, working with Simon, the lyophilization of our virus Supernatant, our virus preps. If you just concentrate on the far left 2 columns, this is an unlyophilized virus, the titer you achieve when doing infections. If you lyophilize the virus and resuspend it, you see you're not losing much virus titer. This will save money on transport and also storage of these viruses. So, hopefully, the data that I've presented convinces you that these simple RNA viruses can be successfully pseudotyped more efficiently if they're plasma membrane budding viruses, due to the different reporters and the stability of these viruses, that they are a very flexible and robust platform for use in a number of different laboratories, while still maintaining this high sensitivity and specificity. I'd just like to finish by thanking long and close collaborators at the University of Kent, Nigel, Simon, Eleonora, Francesca, and Stuart. If you're interested in knowing more about what we do, here are some of our contact details, or I'll be happy to take any questions at the panel meeting.